Hello everyone, and thank you all for joining us today. I'm delighted to welcome you to the Effects of Hypoxia on the Biology of Tumours and Normal Cells webinar. My name is Stephen Robertson and I'm the Sales Director at Don Whitley Scientific. Now, back in October 2019, we hosted the first Don Whitley Hypoxia Symposium. This event brought together hypoxia researchers from all over the UK, providing an opportunity to hear from a variety of speakers engaged in some fascinating work. It was our intention that this would be an annual event, but as you know, COVID brought all such meetings to an abrupt stop. So we hope to run this meeting again in 2022, but in the meantime, we felt this webinar would allow us to bring together the community of researchers with a common interest in hypoxia once again. In fact, it's proven to be widely appealing, with researchers from over 20 different countries attending today's webinar. As you may know, Don Whitley Scientific first started building modified atmosphere workstations over 40 years ago, with the original idea of providing better anaerobic incubation conditions to clinical labs than was available using anaerobic jars. Since then, our expertise has taken us in many directions, but one of the loudest call-ins has been from cell and tissue culture researchers. At first, you just wanted us to improve the incubation conditions so that it better represented the physiologically relevant conditions you wish to mimic. And from that time, our range has grown from 1 to 28 different models in response to your needs, ranging in capacities from 188 litres to nearly 2,000 litres. And you are continually presenting us with new challenges to build wider, taller, deeper machines capable of housing a variety of different equipment. You also wish for the ability to cycle different atmospheric conditions. And now, with the introduction of our latest workstation, we have a GMP compliant chamber. Don Whitley, our founder, was a great advocate for using our chambers as workstations, not just incubators. And he would have been delighted to see that nearly 200 people attending today's webinar are taking such a keen interest. So, on to today. We have 4 fascinating presentations for you. We're covering a wide range of research being conducted under hypoxic conditions. We will hear from our first 2 speakers. Then we'll have a short break, followed by 2 further speakers. And then a Q&A session will be held at the end that will be chaired by our Head of Science, Dr. Andrew Pridmore. Now please make use of the chat function to post any questions you have for our speakers during the talks, which can then be collated and will be asked at the end of the presentations. So on to our first speaker today, who is Professor Karen Kirkby. Karen leads research in proton beam therapy between the Universities of Manchester and the Christie. She has 40 people in her group. She also leads Protons Inspire, which is a European research project that aims to provide world-leading integrating activity for European research in proton beam therapy. Karen has a research grant portfolio of over £28 million from a range of funders, charities and industries. She has over 200 publications, including Nature. And Karen also writes for popular science magazines and is engaged in outreach. Her presentation today is titled Ultra High Dose Rate Flash Proton Beam Therapy.
Thank you for this opportunity to talk to you today. I'm going to talk to you about ultra-high dose rate flash proton therapy. My name is Karen Kirkby. I'm from the University of Manchester and the Christie NHS Foundation Trust. So what I'm going to talk to you about is a little bit on proton therapy with an introduction, then on flash radiotherapy, then look at how we study the flash effect, and finally draw some conclusions. But first of all, a thank you to the team of people that I work with. This is the PRECISE team at the University of Manchester, and you'll be hearing from some of them, and I'm going to be using some of their work in this in this presentation. And I've also got to thank my clinical colleague at the Christie Proton Therapy Centre, who we work very closely with. So what is proton therapy? Well, I think if you look at conventional X-ray radiotherapy, the X-rays deliver their dose in a broad distribution, as you can see here. And what that means when we give that to a patient is, if we're treating a tumour here, you can see that the dose which we deliver from a number of directions comes in, irradiates the tumour, but also there's an exit dose. And similarly, when we deliver it from another direction, and we deliver from multiple directions to try and minimise the damage to the healthy tissue while maximising the dose to the tumour. But you can see there's an exit dose. With protons, because of the way the protons stop in matter, we deposit a little bit of dose as we come in, and then as we come close to what is called the Bragg peak, we deliver most of the dose. Now, obviously, for a tumour, we can't just deliver a single Bragg peak, and so we deliver a range of energies. But if we look at what happens with proton therapy, you can see here there is a sharp fall off at the distal edge, and there's no dose beyond effectively where the tumour is. And you can see, again, we're delivering at multiple angles, but here there's a small amount of dose goes into the healthy tissue in front of the tumour, but beyond the tumour, we don't deliver any dose. So what does this mean in practice? Well, here's an X-ray treatment of a tumour that's located here. And you can see as we're delivering the treatment, most of the dose is going into the tumour, but we're putting a lower amount of dose into the surrounding healthy tissues around that tumour. And, you know, if you can see here, we've got the lungs and the heart and the liver, and we're putting a small amount of dose into that. And obviously any dose is not particularly good for the patient. And so we want to minimise that dose to the healthy tissue while maximising the dose to the tumour. If we now do the same, show the same treatment plan for protons, you can see that there's much less dose going into the surrounding healthy tissue, the organs at risk, and most of the doses conform to the tumour. One of the reasons that the NHS decided to invest £250 million in proton therapy was that dose going into healthy tissue is particularly important that we don't do that for children. This is because any dose gives you a chance of secondary malignancies later on in life. And obviously, if you are 70 and the chance of a secondary malignancy developing 30 years later, that takes you to 100, and you're probably worried about other things at that time. Whereas if you're 10, a secondary malignancy coming along when you're 40 has a much more severe impact. So the NHS developed two centres, one at the Christie in Manchester and one at UCH in London. And these are designed to treat children, but also to treat adults who require complex radiotherapy and where the tumour lies very close to critical organs. So there is a range of indications. What has been built is a 3-room clinical centre. So we have a cyclotron which delivers the protons at 2/3 the speed of light, and gantries which rotate 360 degrees around the patient to deliver the proton therapy treatment. We're capable of treating between 650 and 700 patients a year. As I said, a high proportion of paediatric patients, we're looking 40 or 50%. But we also have the capacity to do a number of clinical trials, plus NHS commissioning through evaluation. The Torpedo trial, which is a head and neck trial, is open now. But 2 new trials, Parable, led by Charlotte Coles, University of Cambridge, is due to open soon, as is Approach, which is led by Louise Murray from University of Leeds. You'll also notice that there's a 4th room here, and this is where we have our research facility, of which I'll talk a bit more later on. This is just showing you some pictures of the clinical centre. You'll notice this is the gantry, and this is the nozzle that delivers the proton beam, which rotates a full 360 degrees round the patient to deliver beams from different angles. The patient also lies on a couch, which has a high degree of movement. which means that we can effectively deliver beams at whatever angle we want. You can see a patient here lying on the couch. This is the gantry. This is the nozzle here, which delivers the beam. And we also have one of these in the research room. This is a picture of the cyclotron, and this is the centre at night. The Christie has been treating patients since December 2018. We've been fully operational throughout COVID. And we've also developed a research quality outcome so that we can actually really monitor what's going on with the patients. It's a very patient-focused service, and you can see here this is one of the rooms where some of the children require anaesthetics, and this is the anaesthesia room. We also do a lot of play therapy, which helps children experience and ask the questions they want about their treatment. This is called our kitten scanner, which is a baby cat scanner, and you can see there's a toy, and this child is talking to one of the play therapists. We also have Proton Panda, which is the mascot for the centre, and every child receives this. And a lot of the Proton Pandas have masks made for them. This is a child who's having treatment for a tumour and requires a mask to make sure that they lie still. And Proton Panda will probably have a similar mask during the treatment. So again, this is a picture of the clinical centre, the cyclotron, with 3 treatment gantries and a room for research. And we've designed the research room so that we can bring a beam through the wall here, and we have a clinical treatment nozzle sitting here, so we can deliver the beam in a manner equivalent to the way it is delivered clinically. We don't have it on the gantry because we can move samples in front of it. We don't have to move it around the patient. And we are developing a second beam line here, which is currently in the design stage. At the end of our beam line A, we put end stations, and I will talk to you a little bit about one of the end stations we've been developed with John Whitley, and my colleagues John, Nick and Mike will talk to you in a bit more detail about that process. But these different end stations are designed to do different experiments so that we've got a wide range of capabilities in the research room. This is a picture of what it actually looks like. This is the clinical nozzle. This is an end station that we used for doing a pan-European radiobiology experiment, and this is the water tank at the end, which stops the beam. Just a quick showing of the hypoxia end station that we've developed with John Whitley, which my colleagues will talk to you about in more detail. And one of the problems with radiobiology experiments is often there's a lot of uncertainty. And what we didn't want to do was put a flask in front of the beam, irradiate it, then have to go in, take it out, pick up another flask, etc. So we've designed an end station that operates at different oxygen tensions, but also has a robot arm so we can do multiple samples. And again, like I say, there will be more discussion of this later on. So going on to FLASH. Interest in this subject really started in 2014 when Vincent Claverdon did some experiments with mice irradiated with high dose rate electrons, and noticed that for those which were irradiated at very high dose rates, typically above about 40 gray, below 20 gray, didn't see the characteristic fibrosis which was normally seen with conventional radiation. So by ultra-high dose rate, we mean above 40 gray, and what appears to be happening with these ultra-high dose rates is it's sparing the normal tissue, but it's still giving the tumour control. This paper has now more than 200 citations. So when we talk about FLASH, we mean about 1,000 times faster than conventional. This FLASH was done with electrons, but FLASH appears to be agnostic of the way it's delivered. And by the way, FLASH is not an acronym, it just means delivered really fast. But FLASH isn't new. There are papers dating back to the 60s, 70s and 80s which also talk about FLASH. But at that time, there was a lot of concern over the tumour control, and the subject went very quiet. There's no real consensus about how to define flash. We know it's delivered extremely fast. How fast? And what is the threshold in time and energy? And are there any other beam parameters that are important? And also, it doesn't induce classical radiation-induced toxicity patterns, but we're not sure about the threshold there. And also the anti-tumour efficacy is still being worked on. Does it work for all tumours? So this looks at the timescale over which conventional radiotherapy and flash operates. And you can see that flash is operating very fast and very much in the heterogeneous and homogeneous chemical and physical chemical stage. And if we actually look at the way that plotted on the way the beams are delivered, I've put here all the types of beam where we tend to see flash against conventional radiotherapy. And we're thinking that the chemistry and the physical chemical processes are very important here. This is a study by Wilson et al., and they begin to wonder whether the quasi-continuous beam used in the cyclotrons for proton therapy is capable of delivering the flash effect. Other studies, they also show, with protons, have been capable of delivering it. So, you know, there was a big question there in 2019 when they published this paper. But the same group have conducted further studies with electrons and protons, and they have now seen the flash effect. And it was very interesting that they had to use low oxygen tensions to be able to see it. So the control of the oxygen tension is obviously really important here. And if we go back to the old literature, again, this is looking at when you see the flash effect. And again, oxygen tension appears to be really important. And more recent studies by Adrian et al., you can see here, we've got flash studies under normoxia, where you don't really appear to see an effect of flash. Whereas when you go to hypoxic environments, you start to see the flash effect. So one of the things that starts to come out is a hypothesis that oxygen depletion caused by flash is one of the underlying mechanisms. And one of my PhD students, Beth Rothwell, has looked into this oxygen depletion hypothesis and developed a model that is a multi-parameter model using a cellular automaton. And it looks at the biological parameters such as oxygen effective diffusivity, oxygen tension, metabolic oxygen consumption, dose rate, radiolytic depletion rates. And one of the nice things of using a cellular automaton type model is that we can actually do this multi-parameter optimization. So this is Beth's model. And what you can see here is she's effectively looked at an oxygen supply from a capillary, and then she's looked at what happens as we move away from that capillary into the normal tissue with both conventional and flash radiotherapy. So if I now play this, you can see here the tissue being oxygenated. And now we can look at the effects of radiation on that. And you can see in the flash, we get hypoxia, generated local hypoxia. And we start to say, is this the reason for the flash effect? And so Beth has applied this model and done over 3 million simulations. And what we're showing here is a multi-parameter plot with the biological, the delivery, and the radiochemical parameters. And the blue lines are showing which parameters you have to satisfy in order to get the flash effect. And you can see it's not just a question of dose rate, it is also the dose and other things such as the effective diffusivity and some of the radiochemical parameters that are important. And when they all come together, we can get a flash effect. And this is a very useful model because it means that we can parameterize the flash effect and basically work out what parameters we have to satisfy in order to get flash. And what Beth then did is she worked with Matt Lowe from the clinical team and said, could she actually put this into a treatment plan? So what I'm showing here is Beth's model in a treatment plan. So we can look locally at what the local oxygen depletion in that tissue is. And the aim of this is so that we can actually build a treatment plan so that we optimize the flash effect in normal tissue while we minimize it in the tumor. Flash has been used to treat a patient, and this is the first patient, a palliative patient, treated with flash, which was, this patient had multiple skin lesions, and one of those was treated with flash. And the results appear to be very promising. There is also a proton flash trial that's being carried out, again on palliative patients with painful extremity bone metastases. And this is being carried out at Cincinnati Hospital in the States, and about 5 patients have currently been treated, and we await the results. We've also run flash in our research room, and this is our first ever flash beam being delivered here. You can see we've gone for that on Twitter. The other thing we've also done is that we've used a flash beam to actually put the Manchester worker bee, which I will show you very shortly. So in terms of flash, it appears to spare normal tissue, and it also appears to maintain tumour control, but we need to do a lot more still. If flash really fulfills the hype, we could deliver it in fewer fractions with less side effects, which mean patients would spend less time in hospital. We could potentially treat more patients, and our patients could return to work and school sooner. So it could be a game changer for radiotherapy, but we really need to understand the mechanisms. And hypoxia appears to be a key parameter for flash. I've shown you some work on the oxygen depletion hypothesis. Obviously, there are other things going on, and reactive oxygen species are being very much implicated in the flash process. But what we have is the equipment and the models to understand the flash effects. And experiments under different oxygen tensions appear to be really important to understand flash, so we need to do more there. And I think watch this space. And finally, I would like to show you our scanning system. So the symbol for Manchester is the worker bee. My team have been working late at night, and they decided to put in a picture of the worker bee and show how good our scanning was under flash dose rates. So here is the worker bee scanned with spots and at flash dose rates. Thank you very much for listening, and thank you again for the invitation.
Thank you, Karen, for that very interesting presentation. And we will hear more in our final presentation about how a robot was housed inside a hypoxic workstation at the Christie, and of the technical challenges of firing a proton beam through the workstation. So, on to our next presentation, which is from Professor Ernestina Schipani. Ernestina is the William Wyckoff Smith Professor of Orthopaedic Surgery at the University of Pennsylvania Perelman Medical School. She earned her MD and PhD at St Anne's School of Advanced Studies, University of Pisa in Italy. Then she did her postdoctoral studies at MGH Harvard Medical School, where she subsequently rose through the ranks and became Associate Professor. She was then first recruited to Indiana University Medical School as a full Professor of Internal Medicine, and next to the University of Michigan Medical School as a full Professor of Orthopedic Surgery, Medicine and Cell Development Biology. Now, early in her career, Dr. Schipani discovered the gain of functional mutations of PTHR1 causes Janssen disease. Next, she pioneered the notion that hypoxia-driven pathways control skeletal development. Her laboratory has established novel principles in broader fields of G protein, coupled receptors, and hypoxia biology. She has published more than 150 papers. She currently studies the role of hypoxia-driven pathways, mitochondria, and reprogramming of metabolism in skeletal development, hemostasis and disease. She has received numerous national and international recognitions. Since 1997, the NIH has funded her research, and since 2005 has been a member of the ASCI. In 2019, Dr. Schipani was the recipient of the prestigious Paula Stern Achievement Award from the ASBMR. In her presentation now, she will talk about how impairment of mitochondrial function can improve survival of hypoxic chondrocytes.
Hi, I'm Ernestina Schipani, and my laboratory is located at the University of Pennsylvania Medical School in Philadelphia. We study hypoxia-driven pathways, mitochondria and bioenergetic metabolism in skeletal development and disease. This is an histological image of a murine growth plate at birth, and within this structure, the growth plate, that is the one that gives origin to most of our skeleton through a process known as endochondral bone development, we have cells called chondrocytes. These cells proliferate. They synthesize a matrix that is enriched in type 2 collagen, form columns which elongate. Then eventually they become post-mitotic cells. They become big, hypertrophic chondrocytes. They now synthesize a matrix enriched in type 10 collagen. They mineralize their matrix. They either die through an apoptotic process, or they transdifferentiate into osteoblast, the cells forming bone. They are invaded by blood vessels and then eventually replaced by bone. Now, developing a growth plate is a unique structure because it's mesenchymal in origin, but despite being mesenchymal, it's avascular for most of its length, though it requires the angiogenic switch in order to be replaced by bone. This is an immunohistochemistry for CD31, that is a marker of endothelial cells. And as you can see, blood vessels are all around the growth plate, but they do not penetrate. So many years ago now, we asked a very simple question. Since the fetal developing growth plate is an avascular structure, is also hypoxic, we used for this purpose the marker of hypoxia EF5, and we showed that indeed the growth plate has an inhypoxic structure. We all know that IF1 is a key factor in cellular adaptation to hypoxia, so the next experiment was to conditionally knock out IF1 in chondrocytes using the Krilox-P system, and the result was quite devastating. So this is a mutant limb lacking HIF in chondrocytes, and this is a wild-type limb, and it's pretty obvious that IF1 is absolutely necessary for endochondral bone development. Why this is the case? This is an histological image of the mutant growth plate and lacking HIF. You see that it is severely misshapen and hypocellular. The center in particular is either severely hypocellular or occupied by abnormal cellular elements with pycnotic nuclei. And when we did the tunnel assay, it was pretty clear that loss of IF1 led to a massive chondrocytes death. So IF1 is a survival factor for hypoxic chondrocytes. What was quite interesting, however, is that the viable chondrocytes at the periphery of the mutant growth plate were much more hypoxic than control. The degree of intracellular hypoxia is the net result of oxygen availability versus oxygen consumption. Over the years, we demonstrate that this dramatic increase in hypoxia of null chondrocytes was not due to my apology, to decrease the oxygen availability. So if it's not decreased the oxygen availability, it has to be increased oxygen consumption. While we were thinking along those lines, was shown that by many laboratories that at least in vitro, a key function of IF1 alpha is not only to upregulate glycolysis and lactic fermentation, but is to suppress mitochondrial respiration with a variety of molecular mechanisms. To prove that this was the case also in chondrocytes, we isolated chondrocytes from IF1 alpha floxed mice. We adenocre in vitro IF1, and we measured oxygen consumption with the seahorse technology. It was pretty clear that loss of IF1 leads to an increased oxygen consumption in both basal state and Indian couple state. So at this point, we know that IF1 is an essential survival factor for hypoxic chondrocytes in vivo, and that in vitro loss of IF1 leads to an increase in oxygen consumption. So we asked then another very simple question: is a stimulation of mitochondrial respiration detrimental for the survival of hypoxic chondrocytes? And to address this question, we looked at the transcription factor TFAM. TFAM is not a direct target of IF1, but regulates mitochondrial biogenesis and activity of the electron transport chain. In particular, regulates transcription of those 13 genes of the mitochondrial genome that encodes protein of the electron transport chain. The other subunits are encoded by the nuclear genome. TFAM also regulates mitochondrial DNA replication. Now, the TFAM conditional knockouts have been shown to be devastating in many tissues that are heavily oxygenated, such as the heart and brain. So we use the PRX1 CRE driver that targets the mesenchymal progenitors that give origin to chondrocytes, and we knock out TFAM in these cells. Now, in vivo. Now, the result was quite interesting. So this is histological images of an of a humerus at embryonic day 13.5 and embryonic day 15.5, stained with a variety of stainings that are specific for cartilage. And we don't need to go into the details. What is pretty obvious, even if you are not an histologist, is that the overall structure of the mutant humerus lacking TFAM it's very similar to the control. It's a bit smaller than control, but overall the structure is similar, both at E13.5 and E15.5. Now, if you look a little bit into details, you see that hypertrophy in the mutant limb is delayed. Now, hypertrophy is the terminally differentiated stage of chondrocytes in the developing growth plate. But proliferation rates are normal within the growth plate. The physiological death at the border between cartilage and primary spongosa is preserved in the mutant chondrocytes. So lacking of TFAM does not alter the overall growth plate structure, does not alter proliferation rates, does not alter physiological death, does not cause any ectopic death, just delays in a modest manner hypertrophy, that is the terminal differentiation stage, consistent with the notion that mitochondrial respiration is more critical for terminal differentiation than for proliferation. Now, interesting, you remember the inhypoxic region of the mutant growth plate of the growth plate is not present anymore in the mutant, indicating that mitochondrial respiration and therefore mitochondrial oxygen consumption are important determinants of this inner hypoxic region. So at this point, we went back to our original question. So is increasing mitochondrial respiration detrimental for the developing growth plate? And for this reason, we created a double mutant lacking both TFAM and IF1, again in mesenchymal progenitors that give origin to chondrocytes. And the result was quite impressive. Now, you see this is a whole mount Alcian blue staining, and you see that the mutant limb lacking both TFAM and IF1, again, it's a bit smaller than control, but overall looks very similar. And clearly that severe abnormalities in shape present in the if null mutant are completely prevented. And so histologically, the loss of TFAM restores the overall structure of the if null limb and prevents that massive hypocellularity of the if null limb and completely prevents the massive cell death. So, knocking out TFAM in the context of the IF1 bring back cells to life. And this is not true also only in the limbs, also in the sternum, that is another target of the PRX1 Cre lineage. We see a picture that is very similar to what we have observed in the limb. So basically knocking out TFAM in the context of IF1 prevents the massive cell death due to the loss of IF. Why this is the case? The current model would predict that everything is due to the ROS. So basically loss of TFAM would increase mitochondrial respiration, this would increase mitochondrial ROS, and this would lead to the cell death. For this reason, we look at mitochondrial ROS and total ROS in our chondrocytes. We isolated chondrocytes from the different genotypes. We cultured them in vitro, either in normoxia or hypoxia, and we measured the total ROS and mitochondrial ROS. We calculate the data both as positive percentage of positive cells and signal intensity. Now, when we look at total ROS, nothing much changes across the genotypes and in these two different culture conditions. When we look at mitochondrial ROS, of course, the loss of TFAM leads to a decrease in mitochondrial ROS accumulation in both hypoxia and normoxia. But what is interesting is what we observe in the if null cells. So, and in the wild type. When we culture, we measure ROS in hypoxic chondrocytes, in wild-type hypoxic chondrocytes, we clearly see that hypoxia decreases ROS accumulation in wild-type cells. And this is prevented by loss of IF. So is IF1 dependent. The interesting thing that if null cells in hypoxia have levels of mitochondrial ROS that are similar to control in normoxia. So since this and this are very similar, probably an increase in mitochondrial ROS is not the reason why if null cells die. So why they do that? You remember that severe hypoxia, virtual anoxia of if null cells in vivo. Well, that severe hypoxia is virtually completely prevented by loss of TFAM in most of the growth plates. In some growth plates there is still a little bit of it, and those are the growth plates in which the cell death is not 100% prevented, indicating somehow a strict correlation between the degree of hypoxia and cell death. And here are the quantifications. And consistent with these findings, by seahorse analysis, we see that the loss of TFAM, my apology, prevents the increased oxygen consumption due to the loss of IF, both in the uncoupled state. And even more interesting, when we culture chondrocytes in vitro, in the two different culture conditions and with the different genotypes, and we look at dead cells by Trapan blue, we see that the only condition in which cell death is increased are hypoxic cells lacking IF1. And this increase occurs in a cleaved caspase 3 independent manner. So if null cells in hypoxia die much more than control. Now, interesting enough, you remember, these cells have levels of ROS that are indistinguishable from controls. So if now we put everything together, this data are consistent with the notion that in vivo, one of the key functions of IF is to suppress mitochondrial respiration, to suppress mitochondrial oxygen consumption, to keep hypoxia in check, and this would allow for cell survival. In other words, when the if is not around, mitochondrial respiration increases, mitochondrial oxygen consumption increases, cells become virtually anoxic, and this is not compatible with life. So we propose that an if-dependent suppression of mitochondrial respiration is required during normal skeletal development to protect growth plate chondrocytes, which are physiologically hypoxic, from lethal intracellular anoxia. The issue is why intracellular anoxia kills the cells. And of course, we look at the ATP and the ATP-ADP ratio by metabolomics. Now, when we look at ATP and ATP-ADP ratio, again, in those different culture conditions, we see that basically this ratio and the ATP levels are pretty normal across the different genotypes, and both in normoxia and hypoxia. And consistent with this, the phospho-NPK-NPK ratio is also normal across the different culture conditions. So this is telling us two things. First, that if null cells are not ATP deficient, either in normoxia or hypoxia. And it also tells us that double mutant cells, DFAM and DIF, compensate somehow the lack of mitochondrial respiration, because now they also have ATP levels and ATP-ADP ratio that are virtually normal. So how do they compensate? So we look at lactate, and it's pretty clear that double mutant cells show an increase in lactate that is virtually identical to the wild-type cells in hypoxia. And this increase happens in a way that is lactic dehydrogenase A independent and is if1 independent, because if1 is not around. So, lack of DFAM and impairment of mitochondrial respiration leads to a compensatory increase in glycolysis and lactate fermentation. And this is consistent with also what is known for other cell types. What mediates this compensation? Probably we have some preliminary evidence that is the dramatic increase in the NADH-NAD ratio that occurs in these cells that could actually mediate the increase. The question is, if null cells are not ATP deficient, why anoxia kill these cells, if it's not energy, at least a steady state? And we wish we had an answer to this question. We don't know yet. We are actively investigating this question, and I'm very, very happy to discuss with you today potential mechanisms and how this could be the case. Thank you so much, and I would like to thank you, the members of my lab who contributed to what we called our TFAM project, and my collaborator Amato Giaccio at Stanford is now at Oxford, and and Ed Lagori in his lab. They have been essential for the success really of the TFAM project. Thank you.
Thank you, Ernestina, for another very interesting presentation. So we'll now have a short break to give you time to refill your coffee cups. But during the break, we'll play a few short videos showing some of the interesting research that's been conducted under hypoxic conditions. Along with those, we'll also show a video that describes and illustrates how we design and build our workstations here in the factory. So I look forward to being back with you in just a short time.
For our research interest over in School of Pharmacy, we use H35 and I2 hypoxia workstation. Our research interest in our group includes exploiting the tumour cell energy metabolism in order to improve gynecological cancers treatment. Gynecological cancers in nature are hypoxic solid tumours. That's why we are interested in performing our experiments under hypoxia at different oxygen tensions, whereby we can actually mimic the environmental conditions that we will see in actual tumours. Gynecological cancers are known to have high glycolysis rate and increased lactate concentration due to the reduced oxygen levels, which we call as hypoxia. We know that increased glycolysis rate and lactate concentration does lead to reduction in survival rate and reduction in overall outcome from these patients. Our work using the Don Whitley's Hypoxia workstation includes measuring profiling of the gynecological cancer cell lines in order to determine their glycolytic capacity under the treatment of the glycolytic stress test compounds that you can get from Seahorse Biosciences. By using these compounds in combination with our cell lines, we have been able to actually identify possible glycolytic inhibitors which would improve the treatment outcome in these patients. We first seed our cells on plates and then incubate these cells overnight for 24 hours or for the duration of the experiment of the treatment that we are interested in. So the next day the samples are transferred through the transfer tunnel into the I2 chamber. This chamber does provide us the environment that we need to do our analysis by using the Seahorse XF analyzer. The unique thing about I2 is that it has no CO2 in it and it has slightly reduced temperature so that the Seahorse analyzer can function. We also have got an internal incubator which allows us to keep the cells at 37 degrees in a non-CO2 environment at 3% oxygen, which is the oxygen level that you can do your metabolic measurements at, for an hour until the media and the cells are equilibrated to the non-CO2 environment. After this, we then follow the manufacturer's protocol provided the XF Biosciences and do actual metabolic measurements on the cells with and without the glycolytic inhibitors. In future, we're hoping to combine these glycolytic inhibitors that we have identified with the help of the dual hypoxic workstation and the Seahorse XF analyzer with chemotherapy and or radiotherapy in order to improve the treatment outcome for the gynecological cancer patients, especially the ones which has got the tumours that are more resistant to the therapies that are available.
We are currently using the Don Whitley H35 hypoxia workstation with the HEPA filter unit. The work that we're doing with the H35 workstation is that we're using it to provide an environment in which we incubate cardiac spheroids, which we form from stem cell derived cardiomyocytes mixed with endothelial cells and cardiac fibroblasts. And this allows the cells to coalesce over a period of time, usually up to about 2 weeks. And then what we do is we allow them then to form a spheroid, and then we can incubate these cells with a range of different drugs and analyze cardiovascular toxicity. Obviously, having the hypoxia workstation is critical for this type of work because we want to incubate the cells in a more physiological environment that they would be exposed to within the human heart. And obviously the hypoxia workstation allows us to generate oxygen levels that reflect the in vivo physiology that these cells would be exposed to. The oxygen concentration that we would use for our application would typically be around 8 to 12% if we were trying to reconstitute normal cardiac physiology. However, for a lot of our work, we actually want to induce periods of ischemia or low oxygen levels, and that's why we chose the oxygen profiling system that goes with the H35, which allows us to pulsate or allow like a periodic low oxygen level of about 1 to 3% that would actually mirror hypoxia or ischemia that would happen within the cardiac muscle. And then we can obviously see how our spheroids or cells respond during that short periodic exposure to the low oxygen. The HEPA filter obviously allows us to maintain sterility during the period of time of 2 weeks to 6 weeks in which we culture the cells, and we don't use antibiotics for our cell culture because we're looking at drug toxicity. So obviously having that sterility is absolutely critical for these longer periods of time. Another feature that's been particularly useful for us is the humidification unit, which allows us to maintain a predetermined level of humidity over a long period of time. Certainly for 2 to 6 weeks, this is absolutely critical so that the cells don't dry out. So an example of our work is shown here with these cardiac microtissues, which are formed from the 3 different cell types that we mix together. And you can see that these pulsate with a regular rhythm. They're about 100 micrometers in diameter, so they allow us to manipulate them and add drugs and see changes in structural and functional parameters. So my experience of working with Don Whitley and communicating with them has been very positive. I mean, what was very beneficial for me was actually to see how the actual station is put together, and it really helps you visualize what type of extras you might need with that system, how ergonomically that's going to work with the particular system that you want. The delivery was very efficient as well, and obviously the service of the unit has been very good. So overall, it's been a very professional package, I would say, that Don Whitley have put together for us.
In our fully renovated facility here in Bingley, we've been able to plan the most efficient production flow model. We've invested heavily in new equipment and machinery. We've improved our warehousing capabilities, set aside areas for customer visits, providing better facilities for staff. We are already seeing the benefits, not least in the number of new products we have been able to launch.
We use Autodesk Inventor as our 3D solid modelling and CAD package to help us work towards productionising the product and finalising the design.
Prototype models are created to test various aspects of the design. We have a 3D printer which is ideal for this process, but we also use traditional model making methods.
We have invested heavily in our machine shop to enable us to create many of our own components and assemblies, making us more self-sufficient. In our machine shop we have CNC milling and turning machines with up to 4-axis control. We are able to manufacture many parts and assemblies in-house. Manufacturing parts in-house allows us to be more responsive, more flexible and accelerate the build process.
Chambers are manufactured to the highest possible standard and heat treated, sometimes referred to as annealing, at least twice during manufacture. This heat treatment process relieves all the stress that would normally occur when acrylic is fabricated, polished or machined.
All our workstations are assembled by hand by our product engineering staff.
Even though these tasks that seem relatively small must be done carefully and precisely, because we know that each component has an important role to play. All our products are designed to work 24 hours a day, 365 days a year, so we have to get each part correct first time. Our workspaces have been carefully created to house everything we need. With the screens we can access materials straight from R&D, diagrams, designs and other materials that we need to ensure a perfect build.
I'm one of the apprentices at DWS. I go to college 1 day a week, and in the rest of my time I help build Whitley workstations.
We take great pride in our apprenticeship scheme, taking on between 2 and 4 engineering students per year.
Every product manufactured at DWS undergoes 120 hours of testing prior to being released out to customer sites.
Whitley workstations are tested thoroughly to national and international standards, including, for example, for electromagnetic compatibility, or EMC. These tests ensure our equipment operates without being adversely affected by the presence and operation of other electrical instruments, and in turn doesn't affect them.
Our on-site laboratories use Whitley workstations on a daily basis. As well as providing a rigorous testing facility, we're able to provide scientific support to our customers for a variety of anaerobic applications.
DWS offers UCAS accredited calibration and validation services. We have also achieved the ISO 9001 Quality Management Standard and the 14001 Environmental Management Standard.
Welcome back everyone to the second half of our webinar. So to our first speaker, who will be Professor Esther Hammond. Esther completed her PhD at the School of Cancer Sciences at the University of Birmingham. Then she accepted a post as a postdoctoral fellow with the Molecular Oncology Group at the University of Cambridge School of Clinical Medicine, before moving to the USA to join the Department of Radiation Oncology at Stanford University. First as a postdoctoral fellow, then as a research associate. She joined the Oxford Institute in 2007, where she is now the Professor of Molecular Cancer Biology. Esther will present her recent work detailing the response of cycling hypoxia and how this can lead to increased hypoechoic activity.
Hello, and thank you for this very kind invitation to present some of the work that has been going on in my lab recently at the University of Oxford. So today I'm going to talk to you about what we've learned about cyclic hypoxia and how this contributes to APOBEC activity in tumours. So before I go any further, although I'm sure it's not really necessary with this audience, I'd just like to be clear about what I mean by hypoxia, and also to make it clear that, of course, it's not just one thing. So most of my lab's interest is focused here in what we call radiobiological hypoxia. And this is a level of hypoxia where oxygen is so limiting that the response to radiotherapy is significantly impacted. So the cells become much more radio resistant, which is shown here experimentally. So this is a colony survival assay where we've irradiated cancer cells at these different levels of oxygen. The red one being what we call normoxia, which is of course not really normal. It's certainly not what cells experience in their tissues of origin. But anyway, we've irradiated the cells at the different levels shown. And as I said, it's a colony survival. So you're seeing the surviving fractions. And as what you can see quite clearly is that we see a significant loss in viability at both 21% and 2%, whereas the response is very different at this radiobiological level of hypoxia. We've got many more cells surviving because they are radiation resistant. And so this is the area of hypoxia, or the level of hypoxia, where my lab predominantly focuses, because we're interested in how cells at this level of oxygen are contributing to tumorigenesis, and of course, if we can identify any weak points so that we could target those cells and therefore improve radiotherapy response. So what have we learned so far over the years? We have discovered that this level of hypoxia, this radiobiological hypoxia, induces a DNA damage response, which I've abbreviated to DDR here. And we've shown this in a number of ways, most simply here through Western blotting for markers of the DNA damage response. So you can see phosphorylation of CAP1, P53, and ATM, and you see these signals at 0.1% oxygen. You don't see these at 2% oxygen, which is sufficient or insufficient enough to stabilize HIF1 alpha. Now, the interesting thing about this DNA damage response is it is a response, but we don't see the actual DNA damage that you might anticipate. So this is a COMET assay showing you exactly that. And that's shown again here on this slide, where you can see that we can see a significant increase in damage if we irradiate the cells, but we don't see that increase in hypoxic conditions. Instead, what we see and what we've characterized over the years is replication stress. So we think it's a DNA damage response to a replication problem that occurs in radiobiological hypoxia. And we've shown that in a number of ways, again, simply by using immunofluorescence for RPA. RPA binds to regions of single-stranded DNA, which accumulate when you've got replication problems. So replication stress leads to single-stranded DNA. Single-stranded DNA gets coated with RPA, and we see these lovely foci. As I said, we don't see DNA damage in these conditions. I showed you that here in the COMET assay. If we use another marker for DNA damage, 53BP1, again, we don't see anything in hypoxic conditions. The situation is slightly more complex with gamma H2AX, which many of you will use as a marker of DNA damage. We do see a response in hypoxia, but I'm sure you can see it's very different to the response to a classical DNA damaging agent, such as neocarcinostatin. The neocarcinostatin has given us these lovely nuclear foci for gamma H2AX. In hypoxia, we see this pan-nuclear staining. So this combined with DNA fiber analysis, which tell us directly that DNA replication is impacted in hypoxic conditions. You can see the replication rates here are much slower than in normoxic conditions. We're confident that there is a replication stress problem in hypoxia. And we've spent a lot of time in the lab studying this from one angle or another. So, what causes the problem? How can we impact replication stress to increase DNA damage? We've asked many of these questions. But One of the questions we've also been focusing on recently is what happens to these regions of single-stranded DNA, and are they vulnerable? And I should point out, I've described to you so far that single-stranded DNA occurs at stalled replication forks. We also now know that it occurs at R loops, which also occur in hypoxia. But I'm not going to talk about that today. I'm going to focus on single-stranded DNA as a whole and ask what might happen to it. And one of the families of proteins that we've become interested in recently are the apobecs. So for those of you who are unfamiliar, this is a family of 11 conserved cytosine deaminases. And so what that means is that these enzymes are able to deaminate cytosine to form a uracil, and they do this in a sequence-dependent manner. So some of you may be thinking, why on earth would we have enzymes that can do this in our bodies? This is a defense mechanism. The apobecs' primary role is to protect us from viruses. So by mutating viral DNA, they can protect us. But the apobecs work, or they deaminate single-stranded DNA. And that single-stranded DNA could occur in a virus, but it can also occur at a stalled replication fork or an R loop. And if you remember, just on the previous slide, I showed you that the hypoxic cells that we're studying are full of single-stranded DNA. So you can see why we started to think maybe the apobecs would be relevant in hypoxic conditions, especially as we know that apobecs are very relevant to cancer in general. So the prevalence of the apobec mutational signature is second only to the aging signature. So the apobec ones are shown in red here. So this is really important to cancer. So this led us to propose this very simple hypothesis. We wanted to know, was it possible that hypoxia-induced replication stress would lead to apobec activity and therefore genomic instability? So we started off by very simply asking what happened to apobec expression in hypoxia. And so we looked by qPCR at the expression of the apobecs that we could detect in our cell line. And sure enough, 4 of them, A3C, A3D, A3F, and A3H, all increased in a hypoxia-dependent manner. And here we've used hydroxyurea as our positive controls because it was known to induce apobec activity, which is relevant because we often use hydroxyurea as a control in our experiments, as it also induces replication stress. Somewhat disappointingly, we didn't see a hypoxia induction of A3B. So this is disappointing because A3B is one of the 2 apobecs which have been linked to this mutational signature in cancer. So it's apobec 3A, which is not expressed in our cells, and apobec 3B, which really drive the accumulation of this mutational signature in cancer. Also, it's important to note that A3B is expressed in the nucleus, and therefore this is the apobec that actually has access to the single-stranded DNA which accumulates in hypoxia. So this was a little bit, this was a little bit concerning, but we thought we would persevere and ask, well, this is just expression. It doesn't tell us anything about what the apobec is doing. So we carried out a functional assay for apobec activity, and that's just shown schematically here, but it's very simple. So this is an oligo with an apobec, a site in it which can be deaminated by apobec activity if present. It's labeled. And obviously, if you get apobec activity, you end up with a smaller product compared to the larger substrate, so largest. So shown here, here's an example of what one of those gels look like, and underneath it is the quantification of several experiments. And just as with the expression, what we found was, as we expect, it went up with our positive control, which is hydroxyurea, but we saw no increase, and in fact a decrease when we put the cells in radiobiological hypoxia. We saw no change in mild hypoxia. So it really looks as though the deamination activity is very much mirroring what we saw with A3B. So this was somewhat disappointing for our hypothesis, but it also raised another question for us. It raised the question of, why is hypoxia and hydroxyurea behaving differently in this situation? Because as I mentioned, we think of we often use hydroxyurea as a positive control because it will induce it will reduce nucleotide level and it will induce replication stress just like hypoxia does. So we got a sort of head scratching moment and thought about, well, there must be something different about hypoxia and hydroxyurea that leads to this APOBEC activity in one and not the other. And the two places we chose to focus were the cell cycle and the presence of DNA damage. So just briefly, this is a FACS analysis, obviously, where we're looking at BRDU incorporation and PI. And if you compare the hypoxic samples shown here with the hydroxyurea samples shown here, you can see, obviously, there's a difference. We can see that DNA synthesis is reduced in both cases, but there is more synthesis with the HU than in the hypoxic conditions. We also found that unlike the hypoxic conditions where we saw no accumulation of 53pp1 foci, just like I showed you on one of my very early slides, we do see 53pp1 accumulating in hydroxyurea. So there is DNA damage in this situation, and that's just shown here. So we've got this difference in replication, we've got a difference in the DNA damage, and we wondered whether these were the factors that were underpinning that finding that HU will induce APOBEC activity, but hypoxia will not. So we started to think about, well, how could we test this? How could we make our hypoxic cells behave more like HU? So how could we think about introducing some damage and maybe increasing replication a little bit in hypoxic conditions? And then, of course, we had a bit of a eureka moment and realized that we'd sort of fallen into the very thing that I cautioned against, which was we'd been thinking about hypoxia as just one thing. Whereas, of course, not only do cells exist at different levels of oxygen, but they also fluctuate between them. So a cell doesn't necessarily sit at one level of oxygen and just experience that. For a number of reasons, cells experience fluctuations in hypoxia. And this is often referred to as fluctuating, dynamic, or cyclic hypoxia. We tend to use the term cyclic. So we wondered whether, firstly, would cyclic hypoxia, would that be more similar to hydroxyurea treatment? And therefore, would it lead to APOBEC activity? So we also knew that there was precedence in the literature for the biological response to cyclic hypoxia being different to chronic or static hypoxia. There are a number of papers which describe different biological consequences to cyclic hypoxia. And importantly, some of these correlate with therapy response. So this is Sam, who was a student in the lab at the time. We reprogrammed the hypoxia chamber so that we were looking at cycling hypoxia instead of just a static level. And these are the conditions that we chose. So the cells are exposed to 2 hours at 0.1%, followed by 2 hours at 2%, and then it starts again. And so we can do however many number of cycles we're interested in at the time. So we set this up. The first thing we had to find out was to be sure that the cells were still experiencing replication stress. So we did that in a number of ways, which I'll just run through. So here is a Western blot showing that CHEK1 is still phosphorylated in cyclic conditions, just like we saw it in chronic. We saw that single-stranded DNA still accumulated in hypoxic conditions. We could see a correlation of two markers of replication stress. So this is RPA foci and that pan-nuclear gamma H2AX intensity. And also, and this is perhaps the most encouraging thing, we saw that as we hoped, we had increased DNA synthesis in these cells. So here, these are the cyclic cells, and here are the cells in hypoxia. And you can see we've got much more BRDU incorporation in the cyclic conditions compared to the hypoxics. So, a huge amount of experiments later, sure enough, in the cyclic conditions, we now see an induction of APOBEC. And that's shown both by immunofluorescence here, where you can see this nuclear A3B staining in the cyclic and the hydroxyurea, and also by Western blot, where again, we've got this increase in A3B here in cyclic, not in chronic, and again with hydroxyurea. Most importantly, this A3B is also active. So this time I'm showing you one of those deamination assays here, and here's the quantification here. So as we expected, deamination increases with hydroxyurea, it decreases with the chronic hypoxia, and it increases with cyclic hypoxia. And although I'm not showing you today, this is A3B dependent. So if we do the same assay after we've knocked down A3B, we don't see this increase. So what's the mechanism? Is it because if we believe that we have made our cyclic conditions are more akin to hydroxyurea treatment, and that part of this is due to that increased DNA synthesis, that increased replication, then if we prevent that, or if we inhibit that, we would predict reduce APOBEC activity. So that's what we did, and we used roscotabine to block origin firing in these cells, and you can see that's been effective. There's a lot less DNA synthesis going on, and sure enough, when we then looked at APOBEC and indeed APOBEC activity, we can see we've got rid of that hypoxia increase that we had previously seen in cyclic conditions. So this is supportive that it is this increased replication in these conditions that has contributed to the A3B. We also wanted to interrogate that other difference that we'd found, which was the DNA damage that we'd seen in the hydroxyurea treated cells. So here we looked for DNA damage in the cyclic cells, and the quantification is shown here, but I think it's perhaps very clear here. You can see that the cells exposed to cyclic hypoxia have got 53 BP1 foci, and importantly, this analysis here shows you that those 53 BP1 foci are in EDU positive cells, so they're in the S phase cells. We also found evidence that the DNA damage was detectable in the cytoplasm. So when we looked at the cytoplasmic DNA marker cGAS, then we could see that this was accumulating in the cells exposed to cyclic hypoxia. So these cells have also got this nuclear damage, whereas it does not accumulate in chronic hypoxia. So we definitely have DNA damage in these conditions as well. Now, some of you might be asking, well, what happens to these cells afterwards? So to investigate that, we put the cells in the cyclic conditions and then return them to 2% oxygen just to see exactly that, what happened. And the interesting thing we found was that the DNA damage persists, so it doesn't appear to be repaired effectively. And perhaps even more noticeably, and you can probably see that very easily on this slide, is that the cells get bigger. That's quantified over here. So these cells are getting bigger and bigger. These images are all to the same scale. Now, obviously, these cells are, I've shown you they've accumulated DNA damage, they're getting bigger. I've also noted down here, they've got wild type P53. So obviously, we are pursuing the hypothesis that these cells are entering senescence. Most importantly, though, can we link this mechanism that we've uncovered, whereby cycling hypoxia is leading to APOBEC activity, can we link that to patient samples? Now, obviously, that's challenging because we don't have a way of measuring cyclic hypoxia in patient samples. So we thought about, well, what have we got and could we use that? And the first thing that came to mind was, of course, the hypoxia signature. So the hypoxia gene signature is predominantly composed of or includes HIF target genes. Now, and if you think about our cycling conditions, both of these levels of hypoxia would lead to HIF1 alpha accumulation and then therefore increased expression of the genes in the signature. And that's just shown, and examples of that are just shown here. So these are 3 HIF1 targets, and you can see they're being increased in cyclic conditions, as you would expect. So we thought that maybe we could use the hypoxia signature as a sort of surrogate for cyclic hypoxia in this case, and use that to ask, was there any evidence of the mechanism that I've described to you leading to APOBEC activity in patient tumours? And the answer was yes, and twofold. So first of all, we found that using the hypoxic signature, the more hypoxic tumours had greater numbers of APOBEC-mediated mutations. So there's a correlation there with hypoxia and that mutational signature that I described at the beginning. And secondly, that there is also a correlation between the hypoxia signature and the expression of A3A and A3B. Remember, the two apobecs that are linked to this signature, and importantly, not to the other apobecs. So we think this provides exciting evidence that there is indeed a link between hypoxia and apobec activity. So to summarize what I've told you today, hypoxia does lead to changes in apobec expression, but notably not apobec 3b. So this is, I'm referring to those experiments where we just put the cells in at 0.1% oxygen. We saw some apobecs increase, but not A3b. In order to get A3b to be induced in hypoxia, we needed to modify our hypoxic conditions to cyclic hypoxia. And this also led to an accumulation of DNA damage. And we believe we and others use the term replication catastrophe to describe this outcome. So this is an extreme version of replication stress where you've accumulated DNA damage, and so is known as replication catastrophe. And we believe that that is what is required to induce apobec activity in hypoxic conditions. We know that that apobec is, apobec 3b is not only induced, but it's active in cyclic hypoxia. We can see from our functional assays. And that led us on to interrogating patient samples. And again, we see clear evidence of a link between hypoxia and A3B expression and activity. So as I mentioned, my ultimate goal is to study this biology with a view to identifying new targets, something that we could potentially inhibit in combination with radiotherapy in order to improve treatment response for patients with very hypoxic tumours. I think it's fair to say that the jury is still out on whether apobec is a good target or not. There are pros and cons to targeting apobec. So it's been known to drive drug resistance and be associated with poor patient prognosis. So of course you'd want to inhibit it. But on the flip side, it's also known to, is associated with the generation of neoantigens, which is beneficial when combining with something like immunotherapy. So I would say that this is still ground for new hypothesis testing, and we are watching this space with interest. So the study that I have shown you was published recently, so you can look up anything that I've missed or not explained very well in this paper. So finally, I'd just like to thank both Don Whitley for this invitation to present our work today, and of course acknowledge the many people who were involved. So the apobec study was begun and really led by a then student in my lab, Sam Bader, who's shown here. He's since graduated and left the lab, and was very, in the later stages, very ably assisted by both Charlotte Simpson and Tiffany Ma. And with that, I would be very happy to take any questions.
Thank you, Esther, for that fascinating presentation. And so to our final presentation, which will be jointly given by Dr. John William Warmenhoven, Mike Merchant and Evan Kitzel. And it is titled, A Well-Defined Problem is Half Solved, Insights Gained from a Successful Project Collaboration. Dr. John William Warmenhoven is a research associate in the Precise Group at the Division of Cancer Sciences, University of Manchester. John has a master's degree in physics from the University of Surrey and a PhD from the University of Manchester on mechanistic modelling in the field of radiotherapy. John's primary research interest is understanding DNA repair through mechanistic modelling, and has developed code that is part of the international TOPAS and Bio collaboration. John has been part of the team that led the commission of the Christie Proton Research Beamline. He is continually working to support the experiments undertaken in the research room and assisting in developing its capabilities. Dr. Mike Merchant is a senior lecturer in proton therapy physics, also in the Precise Group at the Division of Cancer Sciences, University of Manchester. Mike received a PhD in electronic engineering from the University of Surrey, on the topic of beamline design for cell irradiation. From Surrey, he moved to Manchester in 2015 to design the Christie Proton Research Beamline. Mike's primary research interest is the development of biologically augmented treatment planning for proton therapy based on nanodosimetry in the mechanistic modelling of DNA repair. And finally, Evan Kitzel is the design director at Don Whitley Scientific. He has a wide-ranging background in several design disciplines and new product development. He first worked with our company as a design management consultant at the end of the 1980s, then joined the business in 1994. Initially, this was only for 9 months, but he's been here ever since. Evan has been instrumental in the design and function of our range of workstation and plays a key part in any bespoke requests, such as the unit that was designed for Manchester.
Good afternoon. So it's my pleasure to tell you about the development of the Proton Robotic Hypoxia Cabinet at the Christie Hospital. So this was a collaboration between the University of Manchester and Don Whitley Scientific. And to borrow Evan's phrase, a well-defined problem is half solved. Well, we certainly had a well-defined problem at the start, but what we didn't have is a well-defined solution. So we were designing the Proton Research Beamline at the time, and we knew that we needed an end station to go at the end of it. Hypoxia is a major theme of research in radiotherapy in Manchester, so we knew we wanted to irradiate cells under hypoxia, and we knew we wanted to irradiate a lot of cells to reduce those error bars in the experiments. So we had an inkling that we needed some form of automation. So Professor Norman Kirkby and I, armed with a few slides, went to visit the Don Whitley Scientific, Evan and his team, in the Shipley site, which tells you how long this process has been. And actually it was really interesting because Don Whitley himself heard that we were coming and decided to pop in to see the meeting because, well, I hoped he found it interesting, and it was lovely to meet him. So we had a well-defined problem, but we were a very long way from a solution. And our problem was really saying to Evan, would like to fire a proton beam through one of your hypoxia cabinets. But that design quickly evolved with the help of Evan and his team, and we realised from, I suppose, mutually that the design was possible. And from those early concepts, we were able to use that to get funding from CRUK. So we started out with a list of requirements, and really that was to be as flexible as possible so we could meet the requirements of many different types of experiment. So that was using a wide variety of dishes. Always the dishes had to be positioned horizontally because our beamline is horizontal, which presents a challenge in itself. And we knew that we needed high throughput, so we had to irradiate at least 10 samples in a beam-on session over a range of hypoxic and environmental conditions. Of course, part of the challenge was, this is what our proton research room looked like at the moment. We hadn't even finished designing the research beamline at this stage, and we were definitely in the middle of building it. Our room has evolved a little bit since then. Here it is without the proton hypoxia cabinet in it, but you can see the very large scales of the scanning nozzle and the other pieces of equipment, highlighting what a complex challenge this was. And really this design came about because Evan and his team were able to ask us a lot of very detailed questions about what would the robot do, how hot would it get, you know, how big was it, what do the cell dishes look like. The questions were endless, and this was part of a three-party design process that my colleague John is going to describe some of the challenges of.
Thank you, Mike. So as Mike was saying, it was a three-party design between us, Don Whitley Scientific, and Thermo Fisher Scientific. And the first challenge was actually just this collaboration, because one of the partners, Thermo Fisher Scientific, was based over in Canada. So we had to deal with these time zones, and we had to do everything over virtual conference calls, which introduced some lags. But in contrast, Don Whitley Scientific, being only up the road, so to say, were very willing to host. So there was a good, close relationship there throughout the whole process. And again, as Mike alluded to, they were very happy to guide us in their areas where we lack expertise, making sure that the whole process was, or the whole design was integrated to the very highest levels. They were also very willing to learn new techniques, as we'll see later, and used different materials that they hadn't used before. So it really felt like a collaborative effort and not just buying parts off a shelf. I'll just zoom in on some of the specific challenges we went through and how we overcame them. The first one was actually in the design concept. We really wanted high throughput, but we also wanted live imaging. And quite early on, we realized that these two objectives were pulling in different directions. So speaking to our experts, they advised that instead we use a liquid handling solution. So that's what we did. We approached Thermo again, and we got the Thermo Fisher Well-Wash Versa that will allow us to, in an automated way, fix cells after they've been irradiated. Another challenge was that these cabinets are environmentally controlled, so they're sealed, obviously, and we wanted to pass a beam through them. Now, the plastics in there are generally quite thick in order to keep a well-insulated environment and avoid any moisture buildup. But if we were to pass the proton beam through these, the spread that would induce in the beam was really unacceptable to us. It wouldn't allow us to be as accurate as we wanted to be. So, working closely with Don Whitley, we suggested that we could use possibly Kapton, which is used in beam lines essentially to keep vacuum on one side and have atmosphere on the other side. It's incredibly thin, doesn't induce a lot of scatter, but again, we had to come up with a little bit of a compromise in order to keep it thermally well insulated. So Don Whitley suggested we have a foam pad on the other side. And this really demonstrates their willingness to adapt to the customer and really work to their needs. Now, one of the main challenges actually was just that because we're working with Thermo over in Canada, a lot of this was done over CAD. But it's really difficult to CAD the human element, so to speak, to really figure out where the human would go relative to the robot, what space they needed to work in. So Evan at Don Whitley suggested that we use cardboard cutouts and do some physical prototyping. And they really impressed on us the sort of importance and the benefits of physical prototyping. And indeed, they were 100% correct. Because with these cardboard cutouts, we could quite easily have our biologist Amy there go in and move things about, put them where she's comfortable reaching them and interacting with them, and we could quickly figure out the sort of spaces we had to play with. And that leads into the whole sort of spatial arrangement of everything. So the robot had to be in this Goldilocks position. It couldn't be too far to the left or the right, because if it's too far away from the housing unit for the cells, then it wouldn't be able to reach it, stretch too far. And if it was too close to it, then again, it wouldn't be able to bend in on itself and reach the lower units. So we found out this Goldilocks position, but that meant that due to the large, thick cables, a custom adaptation on the side of the cabinet had to be built to house all the flanges for the cables. And then that led us to where do we put the Versa, because it's quite a large bit of kit, and there was very limited floor space. But again, Amy suggested that there are 3 dimensions. Why don't we just lift it up? So Don Whitley came up with this mezzanine idea, and that really helped us. It opened up a lot of the floor space so that we could fit everything in. The other large bit that we needed to fit in there was the hotel, and again, we had to have this reachable by the robot, otherwise it would defeat the purpose. But we also had to have it usable by the biologist to load it and to do any sample prep beforehand. And we wanted this to have about 10 to 19 would be adequate, but ideally towards the 30 sample holding capacity. Combining all of these bits seemed to be a little bit mutually exclusive. So working with Thermo and Don Whitley, we came up with the idea of a movable hotel. So it could rotate and pull out a bit forward for when the biologist was using it, but then it could face the robot and be pushed a little bit back when the robot needed to use it. And you can see here, this really gives us the space, keeps clearance on all the sides so we can get things through the airlock. And you can see me there doing some pipetting. Another aspect that I wanted to highlight was Don Whitley's continued support of us. So after we initially took the end station in, we did some initial tests. We noticed that the front plate, this quite thick perspex, gets really activated. Our limit is 7.5 microsieverts for handling it. You can see there it's already 29 point something microsieverts per hour, and you can see the sort of surprise on my face. So we talked a little bit with them, and we came up with this idea of having a mountable lead glass plate that would fold up and down in front of it, and that's really worked. It's half the activity, which means that we can go and use the cabinet after irradiation much quicker. So before it arrived at us, we did some testing at Don Whitley, and there you can see the sort of whole family of our physical prototyping of the robot and the robot itself inside an MDF mock-up, which we used to test all the paths and clearances before putting it in the actual cabinet shell. And this is important because the paths that the robot takes and just the sheer size of it leaves very, very little tolerance. You can see here how close it gets to everything when it is moving. So Don Whitley were very good in supporting us, coming and installing it and commissioning it at the research room. And you can see there it is in place, ready to be used. And here's a short GIF of it in actual action. So in short, it was a very good project, and we've really enjoyed working with Don Whitley and other partners.
Well, our very grateful thanks to Mike and John for sharing with us their perspective of the collaboration between themselves and our company. And now it's an opportunity for me to just add a few closing remarks to that presentation.
Where you gone? You're out of the box.
Yeah, that's because happened.
I thought it would be helpful first to indicate that the research and development team at Don Whitley Scientific on this project was Dave Boast and myself, pictured on the left of the screen. And then we've had various other people involved behind the scenes and in front of the scenes. So here's just one representative photograph of two of our service engineers installing and commissioning the unit when it was ready for installation at Manchester, albeit just a few days before the first lockdown. So what of Don Whitley Scientific? We started in 1976. We were founded by Don Whitley, hence the name of the company. 3 people to begin with in the spare room and the basement of a small house about 4 miles from here. What's happened in the last 45 years? Well, we wouldn't fit in the small house now. There's 100 of us in 3 countries, albeit sadly Don himself died just 2 years ago, just prior to turning 90. We're still an independent, family-owned and managed business. We have a strong commitment to continuous investment in research and development. We have a powerful blend of science, engineering and design. And although the numbers vary a little from one year to another, about 70% of our production output is exported. We're very proud of that. And we're committed to UK manufacturing. We design it in this building, we make it in this building. Give you a little sense of the research and development capability we have. Very spacious accommodation, very well equipped. We've got all the kind of disciplines you would imagine that we would need internally, but we support that with external specialists and suppliers to enhance and complement our in-house skills. I guess we're arrogant enough to say, particularly where hypoxic workstations are concerned, we've been designing and manufacturing these for about 13 years now. We're arrogant enough to say that if you want to control oxygen to very exacting levels, carbon dioxide to very exacting levels, temperature, including refrigerated temperatures, humidity, and levels of atmospheric cleanliness to a very high standard. If you want any or all of those things, we can design a product for you that will do it. Sometimes when people ask us to incorporate instrumentation inside a product, it would be viewed as something of a compromise. We prefer to refer to it as trying to balance all the apparently conflicting requirements into the best possible solution to suit the customer, to suit the designer, manufacturer, and for commercial reasons as well. How do we do this? We ask a lot of questions, and we quickly seek to make our understanding visible. We turn our understanding quickly into 3 dimensions, sometimes in software, sometimes in prototypes and mock-ups, as you've heard already. And we're trying to gain a sense of the spatial awareness and interdependence of instrumentation, tasks to be performed, labware, and so forth. This will seem odd, but we seek to fail early. And by that I mean we want to learn things quickly during the product development process. It may be that we are heading in the right direction. It may be that there's some unexpected or unforeseen obstacles or difficulties. The sooner we learn those, the quicker we can work through them and come up with a product to suit everybody's needs and aspirations. We also find what customers do and what customers say they do may be different. Equally, what customers say they want and what customers actually need may be different. And so our job is to understand all of the subtleties of this and navigate with our customers through the process so that they end up with exactly what they would like and what they actually need. The English language is quite an ambiguous one and helps when we want to have a little play on words. So we believe that innovation begins with an I. Now the word begins with the letter I, but innovation, the process, begins by observation. We watch as tasks are performed. We take photographs. Sometimes we record things, sometimes we video. We try to really understand what it is that is needed, what it is that's being done, and then we can design products or options and accessories to suit. Let me give you an example. A few years ago, also at the University of Manchester, we were approached to see whether we could either supply from existing product ranges or to design a new one, a workstation that would accommodate an extracellular flux analyzer. It became apparent that none of the products we then had would be entirely suitable, and we began to seek to understand what the requirements were in order to accommodate a device of this kind. So we watched Amy perform tasks in the lead up to tests being conducted within an extracellular flux analyzer. We watched to see what labware was needed, how much space was required, what packaging would need to be disposed of, and so forth. From that work, we were able to develop a suite of workstations interconnected to allow this particular instrument to be used and for the workflow to be ideally handled. We've sold many of these now in various parts of the world. We take a lot of account of ergonomics. It's important for us to be aware that in your own laboratory there will be people of all sorts of different shapes and sizes, let alone in laboratories throughout the world. And so although you can't see the detail from this slide for the brief period it is shown, you can see that we take into account how far people can reach, how far they can see. We're very much interested in wanting to understand the tasks and take those into account, the human interaction, but also the labware and the instrumentation that will also be placed inside our modified atmosphere workstations. Just in the last few months, we've produced modifications to our workstations to accommodate a wide variety of different types of scientific instrumentation. Just a few pictures here to illustrate that point. It's not enough just to run a tape measure over something you'd like to put inside. Refer to the figures that we quote on internal dimensions of a workstation and then say, oh good, it will fit. We need to take into account its weight. It may be necessary to reinforce the floor of the workstation or the trolley upon which it's mounted, as we did with the robot for University of Manchester. Equally, how the product is to be used must be taken into account. Service access, service intervals, connections and connectors, the temperature at which it will most happily operate, the humidity levels it can withstand. Most scientific instrumentation has not really been designed in order to be placed in a sealed box, and so sometimes the heat output needs to be measured, taken into account, and provision made to get rid of that heat. In order to accommodate microscopes, various things have to be done. Please talk to us. If you have a requirement to house instrumentation in a workstation, and the trend seems to be more and more for this to be the case, do please talk to us. The English language benefits from ambiguity, as I indicated earlier. So here's an exploded view of one of our workstations, but our caption can be, yet another Whitley workstation comes together. Please insist on a Whitley workstation. It's a product apart. Thanks very much for listening.
Well, hello everyone. This is Andrew Pridmore at Don Whitley Scientific. Thank you for joining our webinar this afternoon, and thank you, of course, to all the speakers. We're now live in our Q&A session, and you've been typing questions as the presentations have gone along. So I'll now ask them to the speakers in rotation. So first of all, to Professor Kirkby, I actually have a question of my own. I think I understand it now, but being a microbiologist, I wanted to get this clear for my own mind as well. But you suggested that the administration of flash therapy actually causes or leads to localized hypoxia. Is that correct?
Yeah, there's a lot of hypotheses around. Flash is one of those areas that's attracting worldwide interest, and the more people you get interested, the more theories there are. But one of the earliest theories has been localized hypoxia as a result of the flash effect.
Oh, okay, thank you very much. And then Professor Schipani, we have a question here. Are these cells already TFAM/HIF1 mutated, or did you achieve this mutation yourself, for example, using CRISPR-Cas?
Yes, the cells we used in vitro were already TFAM/HIF1 mutated because we isolated these cells from the mutant mice.
Okay, thank you very much. And then Professor Hammond, in cyclic hypoxia, we've been asked, is the number of cycles that you put the cells through a relevant variable to the outcome?
Yep, absolutely. So we currently believe that a significant factor in cyclic hypoxia are the bursts of reactive oxygen species that you likely get when you change oxygen level. So we definitely believe that the more cycles there are, the more of an effect we're going to see. So yes, very relevant.
Thank you. I think that's clear. And then a question for well, Mike and John and Evan. I'll
direct this to Mike and John at Manchester first of all. I think we were wondering, have you encountered any issues with regard to the adhesion of your cell monolayers, perhaps due to the movement of the robot and any vibration, for example, associated with that?
You want to answer that, or should I?
I can have a go. No, we haven't had any issue with any detachment or anything like that. The robot's actually programmed to flip the cells vertically to present it to a horizontal beam at a relatively gentle pace.
Oh, okay, thank you. That's good to know. And Professor Kirkby again, we've got a late entry here, so I'll have to read this off the screen. How plausible is it to use FLASH as a therapy for tissue types where hypoxia is required for normal cell growth and survival? So can FLASH be used in combination with other treatments, and are there side effects with FLASH in these combination treatments? Sorry to.
We're right at the end. Yeah, no, we're right. It's very much. So what I should say is there's 2 clinical trials. Well, the one patient has been treated with electrons, and that wasn't a clinical trial. That was just a one-off treatment to a patient who actually had about 100 skin lesions, and one of them was treated with FLASH. The trial that's going on in the States looks to treat up to 10 patients with painful bone metastases. So effectively using FLASH to reduce, both reduce the tumor and the inflammation as a result. And, you know, it's palliative treatment there. So we don't have any clinical trials that are actually treating patients radically. And although the field is progressing very rapidly, you know, which patients would best benefit from FLASH treatment is a question that's very much out in the open. And one of the things we're planning to do is to get a group of clinicians together to look at what tumours may benefit most from FLASH. And a lot of that is about the local oxygen concentration in the normal tissue, but it's also about the tumours and how we can treat that best. There's a big question about how we are getting the tumor control, and you know, we come into there's theories about symptom reactions, all sorts of radiochemistry. So I'm not giving you a direct answer, and we're definitely not treating any patients with FLASH now. But one of the things that we've got is an opportunity to start looking at the local oxygen environment, and you know, obviously we're doing that in vitro, but we're doing co-cultured cells in the future. So we've got an opportunity to look into it. It's a bit watch this space. You know, I've told you about what's happening and the early stages of it.
Yeah, thank you very much. I think we realise this is very much the cutting edge, so that makes perfect sense. And moving on to Professor Schipani again, does lactate addition restore the viability of HIF knockout chondrocytes upon hypoxic conditions?
We did not do this experiment, but this is a great point. Now, our data would suggest that if lactate is the key, would be the key for reasons that do not depend on ATP production, because steady state intracellular ATP is basically similar across the various genotypes and then the various culture conditions. But again, we did not do specifically the experiment in which we add the lactate to the culture and see whether this rescues some features of the phenotype.
Okay, thank you. And Esther Hammond again, I've asked about the number of cycles. There's a somewhat related question here that is how did you decide the timing of the cyclic hypoxia, and is this similar to the timing that happens in patients?
Okay, that's a really good question, and it is without doubt a limitation of our study in that, you know, we had to pick these parameters. So the way we picked the conditions that we have used to start with is that we were keen to start with that radiobiological level of hypoxia, which I introduced, because we're interested in cells that are experiencing increased radiation resistance. we also, part of our hypothesis was that we would need replication stress to generate the single-stranded DNA. So there were, we had to take the cells down low enough where we thought we might see that. The timing, the 2 hours followed by 2 hours, we searched the literature for studies that have measured cycling hypoxia in tumours, and we felt that that was at least reflective of what has been reported. We also wanted to cycle as fast as we could, and that was what we thought the chamber, where the chamber performed best. We could have obviously made longer cycles, much more easily, but we really wanted to do it as fast as possible. And you're probably going to ask me, how do we know what's going on in the cells? So I should say, we did back that up with the use of an oxygen electrode, where we actually measured the oxygen changes in the dish so that we could see. And obviously, it's not exactly the same as what you program in the machine. You wouldn't expect it to be, but it is, it's obviously very similar, very close.
So are we able to?
Sorry, yes, you asked me about patients. Yeah, the answer is we don't know. And I'm very pleased to be part of a collaboration where we are going to try and start measuring fluctuating or cycling hypoxia to start with in animal tumours, so that we can get more of an idea of what's going on, and so that can then inform our future experiments. So that when I get a question like this, I can say, well, I programmed the machine based on what we saw in a tumour.
Fantastic, thank you. Yeah. Back to Ernestina Schipani. Did you investigate the influence of the TFAM/HIF1 mutation on the radiotherapy response in normoxic and hypoxic conditions?
We should do this. It's a great suggestion. We have not done it, but it's a great suggestion. It's a great point.
Okay, thank you. And is it okay if I ask you another one while here? Which aspects is TFAM? Which aspects is TFAM unable to rescue from the HIF1 knockout? You had suggested the double knockout TFAM HIF1 still left the limbs slightly shorter. Do the residual effects impact the functioning of the adult limb?
Yes, these are great points. Now,
I think, Ernestina, can you hear us? We've lost the volume from you. I don't know if it's the mics. Okay. We still can't hear anything at this side, Ernestina. Well, these events always have this potential, and I think we'll see if it resolves itself, Ernestina, and we'll make sure that we let everybody know the answer to the question.
Do you hear me now?
Oh, we've got you back. Fantastic. Yes.
Great. Thank you. Sorry for that. So I was saying that the loss of the TIF1 is not only a survival factor for chondrocytes, but the loss of TIF1 also delays the differentiation of mesenchymal stem cells into chondrocytes and delays formation of the joints. Now, in the double knockout, we rescue the joint formation, so the joints now develop normally, while the delay of differentiation of mesenchymal cells into chondrocytes, it's not fully rescued. So this is a component of the HIF phenotype that is not fully prevented by the loss of TFAM. About the shortening, the point is that loss of TFAM per se leads to shorter limbs. Because loss of TFAM delays the last step of growth plate development, basically the differentiation of proliferative cells into hypertrophic ones. And the result is the shortening of the limbs. And this shortening of the limbs persists postnatally and persists in the double mutants. So that's the price basically that the mutants have to pay in order for the cells to survive. We are now trying to investigate why the terminal differentiation of cells is delayed by loss of TFAM. Again, it would be consistent with the overall notion that mitochondrial respiration is particularly relevant for terminal differentiated cells. We are trying now to understand the mechanism of why this happens.
Thank you very much. I think there are no further questions coming in on the chat, but I believe, Ernestina, that Esther had a question for you that we were discussing earlier. Is that correct?
Yeah, I didn't realize I'd get this opportunity. So, Stina, I'm obviously very interested in things that happen in an oxygen-specific manner, and particularly down at the anoxic level that you were talking about. So I was wondering about the impact of replication stress, or ATR, ATM, even P53 signaling.
Yeah, you are totally right. And I loved your presentation. I'm totally inspired by it. So we did the double knockout if one P53, because we thought, let's see whether knocking out P53 would affect somehow this cell death phenotype. The double knockout basically looks like the if knockout, but we didn't want to look at the replication stress, and I'm looking forward to talk with you more about it because it's really wonderful. Yeah, so I think that this could be an avenue.
Yeah, and what about the UPR? Because that's the other thing that you'd expect to get involved at that level of oxygen, and also could push the cells into death.
Yes, and UPR can be part of the picture too. We are talking basically many chapters of cell biology here, because as you know, anoxia affects many things, and definitely UPR could be part of it. Definitely could be part of it. Now, what is because basically an uncompensated stress response that would lead to massive cell death. Yeah.
Thank you.
That's excellent. Thank you. I hope I don't surprise you again, Esther, but I have one last question that's just popped up here from a member of the audience. Have you looked at cycling hypoxia in any 3D culture models, and how is A3B expression in 3D cultures, if you've done them?
Yeah, so that is, if the person who asked the question wants to get in touch and give me some suggestions, I'd be very pleased to hear from them, because we've thought about it. We grow spheroids a lot, and I'll admit we've thought, well, shall we put them in the cycling chamber and see what happens? We're really, I think we need the mathematicians to help us work out what concentration of oxygen the cells in different layers would actually experience, and because it's, they usually use a spheroid because you end up with a hypoxic middle and it's very obvious what's going on. But if the oxygen is surrounding in the media is fluctuating, then I'm not quite sure how you model what's going on in the spheroid. So we haven't, because we're worried we wouldn't understand the results. But we've definitely thought about it.
Thank you. Well, that is the end of all the questions now, so thank you very much.
Well, thank you, Andrew, for hosting that session. And it just leaves me to say a few final words, which I suppose we've got to give a very big thank you to all our speakers today. It's been really exciting to hear talks, and I'm sure our audience out there have thoroughly enjoyed it. And we're looking forward to hosting another webinar, and hopefully, as I said, a symposium here at Don Whitley Scientific in the future. So yes, big thank you to all the speakers, and I hope all of you watching the webinar have enjoyed it today. So thank you.